So in the previous video, I say that there are two types of heterochromatin, the constitutive and the facultative. Let's see the first one. The constitutive heterochromatin means it is always in the heterochromatin stage, okay? It will never go back to the euchromatin, okay? Let's say, for example, you kept an old photograph which you thought you don't need, you kept in a what to say, you kept in one old school bag, okay? Then you thought, I don't need the photograph anymore. Then one day your friend says, oh, it's our 20-year reunion, do you have that old photo? Then you will go and get it back. No, you will search and search and search till you find it. So something like that. Even though heterochromatin is your junk DNA, sometimes there are some important genes which arises due to need, due to evolution, due to some changes, something, some disease or some, some sort of mutation happens in the main DNA, and it needs the correct copy. So it will go back and find, so it can revert back, okay? That one is in your facultative, okay? So the opposite of the facultative is the constitutive. Constitute means it's like the constitution, no? You cannot change the constitution. You can change your, what is this, 5-year plans, no? The government can say this 5-year plan we will implement Aadhaar card. Yeah, you can change. Maybe a new government comes, they have another 5-year plan. You can keep changing, no? Another government comes, it will say, no, Aadhaar is not enough. We will make another type of identification. Fine, go ahead. But the constitution, you cannot change it, that the constitution, right? The Constitution of India, that it is made up of this many sovereigns and this many states, this many princely states, all those, that we learned in history, that there will be a president who will be the head of the armed forces, that there will be a prime minister who will be the head of the ministry, then his cabinet. You cannot change that, okay? You cannot suddenly say, oh, I don't need prime minister, I'll become dictator. Go and try. You cannot, So it's like the constitution, you cannot change, okay? It is the same. How, when the constitution was framed, we'll have a president, we'll have a prime minister, we'll have a cabinet, then the states will have their chief minister and his council of ministers, and there will be an election every 5 years, right? There will be some parties formed, then election should be fair, India should be a democracy, and that we have freedom of religion. All these in the constitution you cannot change, okay? So try to correlate between constitution and constitutive, means it will never come back to the euchromatin stage. It will always stay in the heterochromatin stage. Okay, I am speaking too much. I tend to over-explain, no? And I waste time. But as long as you understand, okay. Constitutive heterochromatin remains permanently in the heterochromatin stage. It does not revert to the euchromatin stage. Example, the centromeric region. Okay, it contains short repeated sequences of DNA called satellite DNA. Like the centromere, no, it will always remain as a centromere. Okay, we have evolved in such a way that that junk part will behave as a centromere. Then, looking at the facultative, it is essentially euchromatin that has undergone heterochromatinization. Okay, so just like that old photograph or the old CD or the old cassette, it was useful once in a while, then you got bored of it and you kept it in storage, something like that. So facultative heterochromatin, it was the normal useful euchromatin and has undergone heterochromization, which may involve a segment of chromosome or a whole chromosome. Example, let's see, what is one good example of that is one X chromosome of human female and females of other mammals. Okay, now I don't know if you know this. Yes, females, we have 2 X chromosomes. Okay, this one is X chromosome, this is also another X chromosome. But did you know that one of the X chromosome, it's appearing as a bar body, means it has rolled up itself into a ball. Okay, it's even if you observe the cells of, you can take your salivary cells. If you have a microscope, you can take your salivary, just take a cotton swab, okay, or one of your hair follicles, the cells of your hair, then you squash it on a slide, okay, then you stain it with safranin and you observe under a microscope. You will see one black dot in the middle, means inside the nucleus you'll see a black dot. That black dot is your X chromosome, okay, it's a copy of your X chromosome. Because one X chromosome we already have. Why we need two? No, one copy we already have. This second one, no, it's just a copy of the same. So this is a very good example of facultative chromosome. Okay. So maybe at one point of time, from your mother, no, your mother has X and your father has X and Y. Okay. So in your father, X is as a normal chromosome. In your mother also X is a normal chromosome. Then they fuse into a zygote, and then both X were normal. From the egg and the sperm, they were normal. Then when it reached in the zygote, it realized, oh, already another X is there. Simply one of these will just become into the facultative chromosome. Okay. Okay, let's move on. Oh. I've already told you, so you read this on your own, the differences. Okay. These differences, I think you can read them on your own, no? Because already like so much I have explained from the topic. Okay. Okay, let's look at the chemical composition. Because this chapter is so long, I'm already bored of teaching the same chapter, But I guess for you it's new. There's something new to learn, no? Okay. Okay, so let's look at the chemical composition. Okay. What is a chromatin made up of? Obviously, DNA, RNA, protein. What else? What are your DNA made of? That is simply, chromatin is made up of, DNA, RNA, and protein. Okay, the protein of chromatin is of two types: the histone and the non-histones. Okay, so you must be thinking, yes, we know Miss DNA. Yes, we know RNA. ये protein कहाँ से आ गया? Protein simply is the histone and the non-histone parts. Okay, so those are the proteins in the chromatin. Purified chromatin isolated from interface nuclei consists of about 30 to 40% DNA, 50 to 65% protein, and 0.5 to 10% RNA. Okay, so the DNA is around 30 to 40, protein 50 to 65, and RNA is very small. Okay, but there is a considerable variation due to species and tissues of the same species. Okay, so it may change. Okay, maybe in one species instead of 40 it may be 41% or 42 or 35 the DNA. Then the protein instead of 65 maybe 61, 62. Okay, it changes according to species. The amount of DNA present in normal somatic cells of a species is constant for that species. Any variation in DNA from this value is correlated with the corresponding variation at the chromosome level. Gametes of a species contain only half of the amount of DNA present in somatic cell. The amount of DNA present in somatic cell also depends on the phase of the cell cycle. Okay, it's easy enough. DNA present is constant for a given species. We know that. If there is some change within the species, means something happened in the chromosome, some mutation happened, some problem happened, maybe cancer happened. We don't know, no? That's something that the chromatin changed, okay, that the DNA changed. Then the gametes, they have only half of the amount of DNA. Okay, very easy. I don't think I need to even read that. Okay, so the proteins, we have two groups. They are basic proteins or histones and the non-histone proteins. Okay. So histones constitute about 80% of the total chromosome protein. Okay. So the protein part or the histone, they contain about 80% of the chromosomal protein, and they are present in almost 1:1 ratio with the DNA. Okay. So they are around equal to the DNA. They do not have tryptophan. Okay. Tryptophan is an amino acid. Okay. Histones are heterogeneous class of proteins separable in 5 different fractions. Okay. So when we say heterogeneous, means they are different. Okay. When we say histone protein, doesn't mean only one type. You know, it's not like a box. It's just one type. It's not like a sand or rock or soil. The proteins in the DNA, that is the histones, they are of different types. Okay, they are heterogeneous, means they are mixed. Okay, they are H1, then H2A, then H2B, H3, and H4. Okay, so these proteins, they are different from one another. Okay, H1 histone is rich in lysine. Okay, again, lysine is an amino acid. H2A and H2B are slightly lysine rich, and H3, H4 are arginine rich. Okay, so the H1 is lysine rich, and the H2A, H2B are slightly, and H3, H4 are arginine rich. Okay, lysine, arginine, these are amino acids. So these histones, they are present in all cell types of the eukaryotes except in the sperm of some animal species, where they are replaced by another class of similar smaller molecule proteins known as protamines. Okay. So in all eukaryotes like you and me, we have this. Okay, but some few animal species, they have something a little bit different. Okay, known as protamines. Well, what do histones do? Okay, their role, the function is to organize the chromosome. Okay, remember the beads on a string or the string on a beads, depending on how you say it. It's these chromosomes that organize, that rolls the DNA in such a way that it fits inside one cell. Okay, because the DNA is so long, like so long. Okay, it's like a giant sari, from here till USA. You think about a sari that long. Okay, then you take some rocks and you keep wrapping up that sari. You keep wrapping up around 5 rocks. Okay. Okay, where were we? So histones play a primary function in the chromosome organization, where H2A, H2B, H3, H4 are evolved in the structural organization of chromatin fibers, while H1 holds together the folded chromatin fibers of the chromosome. So the structure is held by H2, H3, H4, and H1, it holds the folded chromatin fiber together. Okay, the non-histone makes up about 20% of the total chromosome mass, but the amount is variable and there is no definite ratio between DNA and non-histone. Okay. There may be 12 to more than 20 different types of non-histone proteins which show variation from one species to the other, and even in different tissues of the same organism. This class of protein includes many important enzymes such as DNA, RNA, polymerase, etc. So the non-histone, non-histone proteins are your enzymes, your DNA polymerase, that very enzyme which does the work of replication. Okay, remember, this is your double-stranded DNA, then the replication fork, remember? Then this DNA polymerase will bind here and it will move and keep on replicating, right? Or if it is a RNA, if we want to make RNA, that very RNA polymerase which creates the RNA, okay, which this molecule, okay, this enzyme is your non-histone protein, okay. So let me clarify again. The histone proteins are those H1, H2, H3, H4, okay, those which create that sort of folded structure of the DNA, okay. While your non-histones are your other things, okay, like the DNA polymerase, RNA polymerase, okay, helicases, all those which take part in your replication, transcription, translation, all those. So I better stop here. It's a long chapter. There's much more to go, but I don't want to overwhelm myself or you.